Hello, welcome to another edition of 5-Minute FRQ, where I take you through an old FRQ on the AP Bio exam in 5 minutes or less. As always, this information is freely available on the College Board's AP Bio website, so if you have any questions, you can look it up. All right, this is number 6 on the 2021 AP exam. The small invertebrate krill species, Thyssinoessa inermis, I think I'm probably mispronouncing that. If I am saying it wrong, I apologize to the krill. Anyway, they are adapted to cold 4 degrees Celsius seawater. Over the past 10 years, there has been a gradual increase in the water temperature of the krill's habitat. A sustained increase in water temperature may ultimately affect the ability of the krill to survive. One effect of higher temperatures is protein misfolding within cells. Krill have several HSP genes that code for heat shock proteins. These proteins help prevent protein misfolding or help to refold proteins to their normal shapes. Scientists conducted experiments on T. inermis to detect changes in the expression of HSP genes when the krill were exposed to temperatures above 4 degrees Celsius. An experimental group of krill was maintained in tanks with 4 degrees Celsius seawater and then placed into tanks with 10 degrees Celsius seawater for approximately 3 hours. The krill were then given a 6-hour recovery period in the 4 degrees Celsius seawater tanks. A control group of krill was moved from a tank of 4 degrees seawater to another tank of 4 degrees seawater for approximately 3 hours and then returned to the original tank. The scientists analyzed HSP gene expression by measuring the concentrations of 3 messenger RNAs, 1, 2, and 3, transcribed from certain HSP genes in both the heat-shocked krill, figure 1, and the control krill. For the control krill, no transcription of the HSP genes was detected throughout the test period. Okay, that was a lot to read. So I have a nice graph here with time on the X and the mRNA concentration on the Y. It's important, so this is a data interpretation question, and it's important that you make sure you're reading the graph correctly. RNA1 are the dark triangles, RNA2 are the open squares, and RNA3 are the open circles, all right? Make sure you read each line correctly or you're going to get it wrong. So before we even look at the questions, let's just look at the trends. So RNA1, the triangles, from 1 to 3 hours, there's no change. From 3 to 6, it goes up. Now be careful, there are error bars here. So between like hours 4 and 5, those error bars overlap. So between 4 and 5, there's no change. But between 3 and 6, it does go up. After hour 6, it goes up rapidly. And at 10, or after 10, it starts going down again. The squares, no change from 1 to 3. 3 to 6, they go up really fast. And then 6 to 12, they go down, and then they just level off. And then RNA3, the open circles, is pretty much doing nothing. Okay, let's look at the actual questions. Identified the HSP mRNA that had the slowest rate of concentration increase. So the slowest rate of increase would be this one here on the bottom, which is RNA number 3. Looking at the rubric, it's RNA number 3. Simple. Okay, there's a lot of scrolling on this question, so I apologize if I'm making you dizzy. Part B, describe the trend in the average concentration of RNA1. So RNA1 is the triangle. So hours 0 to 3, no change. From 3 to 6, it goes up slowly. From 6 to 10, it goes up rapidly. Then after 10, it goes down. And if you look at the rubric, it's what I just said. You can read this yourself, but it's what I just said. Question C, the scientists hypothesized that the heat shock protein translated from RNA1 plays a greater role in refolding than does the one from RNA2. Use data to support the hypothesis. So refolding was the idea of getting the protein back to the original conformation, which is what happens after it's in the hot water. So we're looking at RNA2. RNA2, actually, what's the question saying? Oh, it's saying that RNA1 plays a greater role than RNA2. So look at the period after, once the recovery period. RNA1 continues to go up. RNA2 goes down. So if you look at the rubric, RNA1 is still expressed at a high level after the shock period, whereas RNA2 levels decrease. All right, lastly, I got 30 seconds. Lastly, RNAs 1 and 2 are transcribed from the same gene. Explain how a single gene can produce 2 different products. So this is an unrelated question. This is just content you need to know. It's alternative splicing. You can cut out different introns. You can splice them differently, use different promoters. One gene can give you multiple RNAs due to alternative RNA splicing. Okay, hope that was helpful, and I'll see you guys next time.